Where I want to begin here is to finish up the story from last time on the mitochondria and tell you just a couple of general ideas about the mitochondria. The first thing I'll tell you, and I'll do this from a slide, is that the mitochondria are not really part of our own cells. They're an old capture event from 1.5 billion or more years ago. And one of the ways that that was determined to show that the mitochondria were separate and distinct from the biology of our own cells was they began looking at the mitochondrial DNA. You can see this was an interesting test of that hypothesis, because if the mitochondria were indeed not cellular, not eukaryotic cellular in origin, but rather were of a prokaryotic origin, then they ought to have the kind of DNA and RNA that's common in prokaryotes. And sure enough, when they looked at that, they found that the DNA in mitochondria is far, far more similar to prokaryotic DNA than it is to eukaryotic DNA. And that was one of the major signs, one of the major proofs that this mitochondrion was probably a capture event from some sort of old-fashioned bacterium that was floating around in the ocean at a time when most cells could not use the oxygen in the air. But because of the proliferation of oxygen in the air, because of the evolution of the blue-green algae, of the cyanobacteria, because the blue-green algae were generating oxygen, there were a set of cells which evolved to use that oxygen and make lots of ATP. Those cells are the ancestors of our mitochondria. Now, we'll talk a little bit about this too, because one of the interesting characteristics of the mitochondria is that although sperm have mitochondria and eggs have mitochondria, and although you are the result of the fusion of your father's sperm and your mother's egg, it turns out that your father's sperm got left behind. And all of the mitochondria and all of your cells originated from that mitochondrion in your mother's egg. And this is true for you and your mother and her mother and her mother. It's true that your father's DNA, your father's mitochondria came from his mother. So we now have a matrilineal lineage of the mitochondria. And because we know the mitochondrial DNA sequence, we can look at the rates of mutations in the mitochondria. And by looking at rates of mutations and changes in the mitochondrial DNA, we can get some idea of how the mitochondria, and thereby various mothers and sisters, are related to one another, going back, we think, some 200,000 years. This has been a really important development in asking the question of how people came out of Africa and distributed themselves on the face of the planet. And by looking at various changes in the DNA sequence of the mitochondria, the mitochondrial DNA, that gave us some insights into how people left Africa, migrated up through what's now Saudi Arabia, out into Europe, out into Asia, and then subsequently into North America. And much of our knowledge of how people moved out from Africa and evolved from the very ancient groups in Africa, how that happened, is largely a result of understanding of mitochondrial DNA. And I won't say too much more about that. What I want to emphasize today is this idea of the cytoskeleton. The idea that there are filamentous proteins inside the cell. The idea that these filamentous proteins are polymers of other proteins. But they're not just random proteins. We'll find that microtubules, which are about 24 nanometers in diameter, but can be hundreds of micrometers long, that these microtubules are polymers of a particular kind of protein called tubulin. That one of the characteristics of these tubulin molecules is that they self-assemble one with another, but they don't self-assemble willy-nilly. They self-assemble in such a way that they provide an endedness to the molecule. They're always oriented in the same way. And because of this orientation, one can say that the molecule has a kind of endedness, an assembly end where we add tubulins, and a disassembly end where we take the tubulins away. This becomes important because it gives us a kind of directionality to the molecule. That directionality of the molecule becomes important when we begin thinking about the motors that are now going to use the microtubules as rails along which they travel. Now, yesterday we blithely talked about moving vesicles from place to place inside cells. what I want to emphasize today is this idea of how we use these molecular motors to attach to vesicles, and then by using the molecular motors, we can walk in one direction or the other along these oriented structures, the microtubules. We're going to see that there are two major protein types. There are two major motor types. That is one called kinesin, and when you see kinesin, always think plus end directed. That gives an orientation of the microtubule. This tells us the kinesin is going to move vesicles only in one direction, toward the plus end, toward the assembly end of the microtubule. But there's a cousin molecule, another molecule called dynein, which moves the vesicle in the opposite direction because it walks along the microtubule in the opposite direction. So now we have directional movement that's been determined by the characteristics of the motors, either dynein or kinesin. What these motors have in common is that they are ATPases. And what we mean by an ATPase is they will bind to adenosine triphosphate, and then in rapid, short order, enzymatically, quickly convert that adenosine triphosphate to adenosine diphosphate in an organic phosphate. They're clipping off one phosphate from the three. When that happens, that then changes the structure. There's a conformational change in the structure of either the dyneins or the kinesins, and allows these molecules now to step, one step at a time, along the tubulins, walking along the microtubules from one end to the other. These occur really fast. These changes occur about something like 500 times a second. The other character that we're going to see as being an important transporter substrate molecule inside all of our cells is one called filamentous actin. Again, it's a protein polymer. Filamentous actin is assembled from individual actin molecules. Each actin molecule is a molecular weight of about 40,000, and you plug them together just as they were pop beads. You sort of string them together and you make a polymer. But again, there's only one way you can string them together. There's only one way that you string them together such that there's an assembly end to which you add a new actin monomer, and there's an end from which it falls off. Now, under normal circumstances, there are other proteins which will stabilize the structure of the actin so that we have now this linear structure with an end, a plus end and a minus end. And again, we have a motor. Now, this is a motor you may recognize. This one's called myosin. This is the myosin, and it's the myosin-actin interaction which we see in muscle, and we'll spend a lot of time on that, which permits muscle movement. It allows us to walk about. It allows us for our muscles to be active. But here, we have a slightly different story. These are older forms of myosin and a slightly different form of actin. They tend not to make the great big bundles we'll see in muscle, but nonetheless, they obey the same rules. That is, there's a substrate of actin and a motor of myosin. This is an ATPase, and this motor of myosin can attach to a vesicle in this case and now move vesicles along the substrate, all using ATP. Now, if there's time, we'll see that these actin and tubulin molecules, the actin filaments and the microtubules, are not randomly distributed inside the cells. They're regionally organized. They're found in different neighborhoods. And for the most part, we find the actin out along the edge, adjacent to the plasma membrane. This is called cortical actin. This is called the cortex of actin. So most of the actin is around the outside. I'm sorry, let me do it this way. The cortex of actin is lined up right along the cytoplasmic side of the plasma membrane. It's a very thin layer of that right inside the plasma membrane, whereas in the core of the cell, toward the middle of the cell, you find bundles of microtubules. This has led people to think there are two kinds of transport here. There's a transport on the substrate of microtubule, and then perhaps a local transport, bringing the vesicle right up to the membrane using the cortical actin. And we'll talk a little bit, if there's time, about how that gets done, and then perhaps we'll get time to talk about DNA.